Ladies and gentlemen, good morning.
Okay, we
you're
are
so
here
welcome
live for
the
Nobel
announcement
Prize
of
Week
this
Day
year's
1,
Nobel
which
Prize
is
in
medicine.
Physiology or
Looks like the
Medicine.
presentation is starting.
My name is
All right, let's
Thomas
go.
Perlman, and I'm the
2:30 a.m.,
Secretary
baby.
General of the Nobel Assembly.
Okay,
I
here we
will
go.
first read the announcement in Swedish, followed by English. We will then, as usual, present some background to the prize and open up for questions.
Okay, here we go.
Yep.
Nobelförsamlingen vid Karolinska institutet har idag beslutat att Nobelpriset i fysiologi eller medicin år 2025 skall delas lika mellan Marie Brunkow, Fred Ramsdell och Shimon Sakaguchi
Okay.
för
it was a
deras
crapshoot
upptäckter
anyways.
rörande
So the predictions we did yesterday.
periferial.
So the predictions are wrong.
Okay.
The Nobel Assembly at Karolinska Institutet
here we
has
go,
today
finally.
decided to
Civilized
award
language.
the 2025 Nobel Prize in Physiology or Medicine jointly to Marie Brunkow, Fred Ramsdell, and Shimon Sakaguchi for their discoveries concerning peripheral immune tolerance.
Okay.
Okay.
Here
Peripheral immune tolerance.
are the three laureates. Marie Brunkow was born in 1961 and received a PhD from Princeton University in the United States.
Whoa.
The work for
Let's
which she is awarded was
go!
performed at a biotech company, Celtech Chiroscience in Bothell, Washington. She is currently a senior program manager at the Institute for Systems
Oh, wow. So
Biology
it's not even an
in Seattle.
academic institution.
Oh, interesting.
Fred Ramsdell was born in 1960 and received a PhD in 1987 at the University of California in Los Angeles. The work for which he is awarded was
Princeton,
performed
you
at the
see.
same biotech company, Celtech Chiroscience. He's currently a
Nice.
scientific advisor at the company he himself founded, Sonoma
Can't believe you got the double.
Biotherapeutics in San Francisco and in Seattle. Shimon Sakaguchi was born in 1951, earned an MD in 1976 and a PhD degree in 1983 from Kyoto University in Japan. The work for which he is awarded was initiated at Aichi Cancer Center Research Institute in Nagoya. He's currently a distinguished professor at the Immunology Frontier Research Center at Osaka University.
Okay. At Osaka University. Okay, so we got two private people and Osaka University. I think we need a wardrobe change.
Yeah, no, I mean, what?
Yeah, that's crazy.
Okay.
All right, I'm going to do some research.
And we'll dive in.
Yeah, and then we'll be right back.
We'll be right back. Hello, Internet. This is your captain speaking, Lester Nare, joined as always by my co-host and our resident PhD, Krishna Choudhary. We are now doing a deep dive on day one of the Nobel Prizes. We had the Medicine Award for Peripheral Immune Tolerance just go out. As you can see, we have a little bit of Princeton representation. 'Cause
Yeah.
we are back on the board with one of the Nobel winners who just won, being a PhD at Princeton.
Yeah.
But let's dive right in. Peripheral immune tolerance, just one in medicine. Why? Like, let's start with why this prize matters.
Yeah. This prize matters because it's effectively, if you go to slide 2, it's the military police for our body. Okay? The military you can think of as the immune system. They're the ones that are fighting the invaders, right? But every once in a while, some people might act up, okay? And then what ends up happening is you need to control parts of the immune system that might be attacking the self. And that's where these regulatory T cells come in, okay? And that's why I think it's kind of like the military police. And these three Nobel laureates in slide 1, Shimon Sakaguchi, Marie Brunkow, and Fred Ramsdell, they are responsible for doing a lot of the research that elucidated this system of regulation of our immune system. Basically answering the question like, why doesn't everyone have an autoimmune disorder? Okay?
Right.
And that sounds like kind of crazy, but it'll become a non-trivial problem once I get into some of the details about exactly how our immune system works, okay? So with the immune system, there's two different kinds of basic immunity that we have. So in We've got the innate immunity, which is what you see on the left over there.
Yes.
That's stuff that doesn't require learning. That's in our genetics.
Right.
We're wired to recognize viruses. We're wired to recognize bacteria. We're wired to recognize outside pathogens that are coming in, okay? Then there's something called adaptive immunity. These are our T cells and our B cells,
Right.
okay? They're specialized to have a kind of memory of stuff. They're specialized to really recognize things at a molecular level. And these things are slower, and it involves a lot of different molecular mechanisms that we're going to try and get into. What we're really going to focus on are something called T cells.
Okay.
Okay. T cells, they're a type of white blood cell. Our blood has basically 3 types of cells. We've got the red blood cells, which carry oxygen. We've got platelets, which do a lot of the clotting and like tissue repair. And then finally, we've got white blood cells, which are our immune system, right? And the ones that we're going to focus on today are called T cells. They develop in the thymus, which is an organ of our body, hence the name T cell.
T cell.
Okay. There's 2 types of T cells. There's the helper T cell, which coordinates an immune response. It doesn't actually do the killing,
Yep.
but it like tags stuff and then makes other like B cells come in, or macrophages come in. Macrophages like big eaters that like come in and like actually like eat things
Yep.
and digest it,
Yes.
like within the cell.
Yes.
And
So
then
they're like scouts, right?
yes, the helper T cells are exactly like scouts.
Yep.
Yeah.
Okay.
And then you've got your killer T cells, right? And the killer T cells, like the one you're seeing over there, that's attacking the smaller one is the killer T cell, and it's
Yes.
taking on this giant cancer tumor cell,
Okay.
right? And it's attacking it. So killer T cells can kill infected cells. They can kill cancerous cells directly. And the mechanism by which they understand whether something requires killing or not
Yes.
is the focus of this problem.
Got it. Okay.
Does that make sense?
Yes.
Because imagine, right? You're a cell. The senses that you have are just lock and key. That's like at the cellular level, you're worried about concentrations of certain molecules. Like, how many of this molecule do I see around here? Is that a red flag? Or on the other hand, like, you go to another cell. How do you know if that cell is healthy or it's been taken over by something? Right. And to get into that, we're going to have to go into exactly how these T cells actually find, which one is wrong, which one is right.
How does our first line of defense
No. Yeah.
what to go after?
Yeah. And effectively, like, in a normal society, the police come over and they check your ID. These guys are doing kind of the same thing. Okay. So in slide 5, you'll see this is from the Nobel Prize Committee. They release these like, things for the press.
Yes.
To sort of explain and understand. It's a really good cartoon. So on the left-hand side, you've got a healthy body cell that's been infected by a pathogen, right? That's the stuff that's coming in. And what these cells do is they've got these receptors on the outside of their surface that actually take whatever is inside the cell and display it.
Got it.
It's like an ID card for everything that's inside your cell. Okay? Every little protein that's been degraded or like broken down, they're going to take little parts of that protein and they're going to put it out.
Got it.
And then what the T cell does is they've got receptors that actually come in and bind. And if one of the receptors matches, like it does over there, then it's like, oh,
For that
that's
T
bad.
cell, that's sort of center screen.
Yeah,
Yes.
that's sort of attached to that
Yes.
receptor. That T cell has recognized something that is quite bad.
Yes.
And so it's going to elicit a immune response. That's that lock and key model that I was talking about.
Yes.
Right?
Yes. I think there's a literal scene in the movie Osmosis Jones that walks through T cells going and checking ID.
Oh, really?
Like, as the metaphor for
Dude, that's literally what this is. Yeah. And so the way that the ID check happens is if you go to photo 6, this is a picture of the human leukocyte antigen, okay? And this is the protein that is responsible for taking whatever is inside the cell and displaying it. So that red part that you see at the very top,
Yes.
like right over here,
Yes.
that red thing is maybe it's a viral fragment, or maybe it's a part of our own DNA that or our own protein complexes inside. What they've done is they've broken down the proteins and they display them in little tiny segments outside. Okay, and the T cell's job is to come in and try to figure out if that little fragment is self or not.
Yep.
Understood?
Yes.
Okay.
Yes.
So this human leukocyte antigen, it's part of the MHC major histocompatibility complex. It's found in like, animals and so on and so forth. This is the thing that's actually involved in like mate selection. So like, have you heard about that research where it's like people with different immune systems smell better?
Yeah.
And
yes.
that's why you're like more attracted to them. That's this stuff.
That's interesting. I
Okay,
know
that's
exactly what
the
you're
human leukocyte antigen. Okay,
got it.
like everyone has different versions of this antigen, and really only like your identical twin has the same one. Okay, and the more different you are, the better it is for the kid.
Because the idea is there's less of these potential threat vectors that you are going to be susceptible to because you're so different.
Yeah, exactly. And so like you have like a broader sort of
immunity.
And the other reason, like organ transplant rejection, a lot of times that happens because your HLA, this antigen complex, is different, and so the organ is like, that's not me. Right?
That makes
like,
sense.
and I don't want that as part of this body. And so people who are actually studying this organ transplant problem, they won the Nobel Prize in 1980, Jean Dassault and George Snell, for discovering this human leukocyte antigen, this like idea of like these proteins that actually take what's inside the cell and display it out. And the fact that they're different is what causes all of these rejections. Okay? So that was our first clue as to like how this stuff works. Okay?
Understood.
So now we've figured out how cells in our body display their ID.
Right. In order for the T cells to be able to identify whether or not it's something that they need to deal with or not.
Yeah. Now, the next part of the clue is, how did the T cells, which are the police, or the military in this case, how do they decide whether that ID is okay or not? Okay? And so this
one thing is the display of the ID.
Yeah.
The second thing is like running it through is like actually scanning it to be like, are you in the database?
Are you in the database or not? And that's where T cell receptors come in. Okay? So T cell receptors they look like that. Okay? They're the ones on the outside, and you can think of this as the key.
Got it.
If the HLA things that we were talking about earlier, where the cells are displaying it, that's the lock. Okay? And these keys have to match the lock. And if they match the lock in a bad way, then that means you're a bad person. If they sort of match it in a good way, then you're fine. Right?
So the idea is these T cell receptors are sort of protruding, looking to be able to fit into the lock of a matching, person on the bad list. And if that then happens, then they know we need to now have our appropriate immune response.
Exactly. Yeah. And it's actually kind of an insane thing to think about, right? Because as a T cell, you're going to have to come up with a bunch of different keys.
Right. For
and
all
that
the
means
stuff.
for all of the stuff that's out there. And if you want to have a bunch of different keys, you want to have a diversity. This is in the protein. You want different shapes in your protein. That means you need different sequences of amino acids. You want different sequences of amino acids. That means you need a different sequence of DNA creating that amino acid. And for the longest time, it was like a really big challenge to figure out, because we have like something like 10 to the 15 different receptors. Okay, to code for 10 to the 15 different receptors, you would need like 10 to the 13 amino acids at least, or 10 to the 14, right? Because it's 3 for 1. So I'm doing some really crude math here. 10 to the 14 different sequences of DNA. That's a lot of DNA.
Right.
Okay? That's larger than our genome.
Right.
Okay. So how does that happen?
How are we actually storing the memory and facilitating the creation of the T cell receptors in this wide, this massive scale that is necessary for us to survive in the way we survive?
Exactly. And if you go to photo 8, that's a pretty good depiction of it. Like on the right hand side, you've got all these different shapes, right, that the T cell receptor can be.
Okay.
Right? And it's trying to generate all of these different shapes. And the challenge is, how do you generate so many different protein sequences without blowing up on the genetic side of things?
Right, correct.
Right?
Correct.
And that was made possible by something called VDJ, Variable Diversity and Joining Locuses. They're different gene segments. And what ends up happening is the cell mixes and matches. So it's like you have like 10 different pairs of socks,
Yeah.
20 different shirts, 20 different pants.
Yes.
And now you can come up with a combinatorially much larger outfit.
Yes.
Right? That's exactly what these T cells are doing.
Got it.
Okay? And this process was discovered by Susumu Tonegawa, and he won the Nobel Prize in 1987. He's now at MIT. He's doing actually optogenetic stuff, which is what I had
which has
slated.
as the prediction.
He's actually chasing his second Nobel, I'm pretty sure.
Oh, okay,
He's
yeah.
trying to, he does like optogenetics on the hippocampus, which is the Center for Learning and Memory in our brain. So good luck to him on that. And then the last part of it was, Tonegawa figured out how to make all of these different proteins, right? All of these different shapes
Without blowing up,
right?
The genetic base.
The final key was in 1996, they won the Nobel Prize, Peter Doherty and Rolf Zinkernagel, and they were the ones who showed that actually that human leukocyte antigen, the part that was displaying the ID, that's crucial for the T cell lock and key model. The T cell isn't just locking in on everything.
It's
right?
doing it.
It's doing it specifically on the stuff that this thing is putting out.
So
and displaying.
Instead of doing no-knock warrants at every door, they check the ID at the door and only do the bust when there's a match there, which then decreases the volume of the activity itself.
Exactly. Yeah. Now you can be a lot more specific. You can manage the whole.
You can actually scale to a complex
organism like us with like billions of cells, right? Yes. So that's the background behind how T cells do their job if they're doing it correctly.
Right.
Okay?
Okay.
Now, some T cells are inevitably going to recognize your own body. Okay? Because these guys are making, as I said, 10 to the 15 possible receptors, right? Some of these receptors are going to match to like the stuff that we have, right? How do we make sure that those guys don't just like go off? Right. Because each T cell is expressing a specific shape.
Yes.
Okay? Each individual T cell is expressing a specific shape. So what we basically want to do is the ones that are recognizing our body, we should just get rid of them. We should tell them to commit cell death.
Yes.
And then we don't have to deal with that anymore.
Got
so
it.
that first line is called central tolerance. That's the first filter.
Yes.
It happens in the thymus, okay? So these T cells, what they do is they can bind, this is photo 10, they can bind strongly to self-proteins. And whenever they do that, the process that happens in the thymus when we're very, very young is those T cells go off and they don't proliferate to the rest of the body.
Okay.
The filter is not perfect though, and there's some that get through. Okay, that second line of defense, that is what this Nobel Prize is about.
After that first sort of purge, there's still some remaining leftover T cells that recognize our own bodies
as invaders.
As invaders, incorrectly,
yeah.
And so now there's a second after the first filter, which you just talked through. There's a second line of defense.
Yes, yeah, and that's photo 11. If you go there, these are called regulatory T cells. So on the left-hand side, what you're seeing is, this rogue T cell that's misbehaving, that's recognized our own body and a protein fragment in our own body as something that needs to be taken care of. Now comes a regulatory T cell, and what that does is it's going to suppress the immune response of that incorrect T cell. Okay? And this regulatory T cell is what stops the autoimmune response from happening. Autoimmune meaning it's an immune response on yourself.
It's almost like internal affairs in those cop movies when the cops do something bad and they come in and they investigate their own people to make sure that it gets taken care of, and you remove the people from the force that are problematic.
And so if only that actually happened, but that's exactly what's going on.
Okay.
It's like you've got like the ones who regulate the cops.
The cops,
right?
Yeah.
And if they do a good job, like in most of us, we don't have those.
Yes.
Right? And so mostly it works. And where it doesn't, we need to figure out what's going on with the regulatory T cells and if there's ways to prevent that from happening.
So we have our first filter, and then once we get past the first filter, there's still some remaining T cells that are attacking our own body. So then regulatory T cells come in to suppress that immune response. But that doesn't happen 100% of the time still, even at that second layer.
Yeah, and so now there's ongoing work even today. Like I was looking today at when I was like researching this stuff. Even today, there's papers coming out about how do we make this system better. But the first step is always understanding how the system works, and that's what our 3 Nobel Prize winners did. Was just figuring out that the system existed in the first place. Discovering the fact that there are such things as regulatory T cells that are uniquely different from the rest of the T cells, from helper T cells, from killer T cells. This is a totally different class of immune cells that come out of the thalamus, right?
So you have the ID system, you have the T cells that are the ID system, you have the T cells that are going out and actually doing the work. And then now there's this third bucket, which is the police of the police.
Police of the police, exactly. Yeah, and these guys are the ones who like solved it. Okay, so now we're gonna get into actually what these 3 researchers did. We're gonna start with Shimon Sakaguchi. He got his PhD at Kyoto University, and then he started getting really into these autoimmune disorders. So the first thing he did was figure out that actually if I lesion the thymus of an animal in the lab, then the animal develops an autoimmune disorder. Okay? So the thymus is key. I mean, this was already known, but now you've got sort of test bed. Okay? And the key thing that he did was he injected a fraction of immune cells, like over there. On the top row, you see just you lesion the thymus, the guy's sick. But if you take a fraction of immune cells from a healthy mouse and you inject that into this guy who doesn't have a thymus, he's going to be fine.
Interesting.
So what you've done is there's something in the mature immune cells of a healthy individual that when you transfer to someone who does have this no thymus defect, he's going to be fine, right? So what you've done is transfer this tolerance, right? You've transferred this mechanism. Okay. And this was back when like they didn't have like monoclonal antibodies. They didn't have any of this stuff. So they used to use like blood serum to basically classify like what was in the thing that I injected, which is like very coarse grained, right? Now nobody uses it, but back then that was cutting edge. And so you just knew that in whatever cocktail that I injected it, there was something magic, okay? Then came the introduction of monoclonal antibodies, right? And with monoclonal antibodies, now what I could do is I could start identifying a subpopulation of T cells that have a specific thing, like a specific surface marker, because the monoclonal antibodies will then go and attach to that surface marker, and then you can say, oh, this thing has the CD4 plus, or the CD425. CD is just like a naming convention for like the proteins that are on the outside. And the number is just like the number in which that it was discovered. So you can now start saying that this specific T cell that mattered actually had this very specific type of protein. And so it's different from all the rest that we've been looking at, right? And he started calling these regulatory T cells or Tregs, right? And without these cells, the mice developed autoimmunity, but with them, they were fine, right? And he finally published his paper in the Journal of Immunology. This was the first sort of seminal paper. In 1995. Okay. The first one with the serum was published in the 1980s. Everyone was like, yeah, probably. But at the time, you know, that's the best you can do. Now we've gotten to like this resolution of like, now we've got cells. All right. So
there's a level of higher fidelity. To get to the answer.
Exactly. And so fine, now you've got these cells. We still need a little bit more convincing, right? You got to get to the genetic level to really see if there's something that is changing at the gene level that is expressing a difference between these regulatory T cells and the normal T cells. Because even normal T cells, like the conventional activator effector T cells, they also have CD25, which is this marker that he had identified. Sure, fine, it's like a little bit lower density, but it's still there, right? So it's not as convincing. Right. And so
what we're saying is, what is the actual mechanism that allows the differentiation in when these T cells are, you know, when they get created? Like what makes it know I'm a helper?
I'm
versus a killer
or
versus a regulatory. Is that
exactly like the courses that they took in the training academy?
Right.
But what exactly did they read?
Right. How did they get to? Their
exactly.
placement?
You know what I mean?
Yes.
So that was Shimon Sakaguchi. he had sort of come into the foray. Now we're going to get to our next two Nobel Prize winners, Marie Brunkow and Fred Ramsdell. Their story is pretty interesting. So actually, to get to their science, we first need to talk about 1940s Oak Ridge National Lab. I think you've got a photo of that there.
Yes.
Yeah, that's where we enriched uranium for the atomic bombs.
Yes.
And there's a lot of radiation there. And in the 1940s, researchers found a mutant mouse strain in Oak Ridge that had scaly skin, swollen lymph nodes, early death. And, as scientists do, they're like, oh, let's study this thing.
Right.
Okay. They called it the Scurfy mouse strain. Okay. They called it the Scurfy mouse strain. And what they noticed was only male mice developed this disease.
Okay.
Of like having this like sort of autoimmune type of disorder. Right. And so if you look at photo 17, if only male mice display this disease, but females are fine, that means that it has to be an X-linked disorder. X-linked meaning that whatever genetic thing is making this thing happen is happening on the X chromosome, right? Because females have 2 copies of the X chromosome. So if one of them is defective, the other one is still going to print the correct protein and you're going to be fine. But if you're an XY and this thing is defective, then you're just totally screwed.
Okay,
right?
That makes sense.
So they figured out that it's got to be on the X chromosome. They did a lot of different tests to figure out, and they narrowed it down to 500 base pairs. Okay. And their job, what Brunkow and Ramsdell did, they were these basically gene hunters at Celtech Chiroscience. Marie Brunkow had finished her PhD at Princeton. We got another one, baby.
We got it. We're on the board.
And then Fred Ramsdell at UCLA, which is where I went for PhD. So that's kind of cool that today both my institutions.
I was going to say, but both
they got a +1, baby. Yeah. So after they're done with that and they're done with their postdocs, they joined Celtech Chiroscience, which is I think it's kind of like a bioscience startup in Seattle at the time. And what they want to do is, if you go to number 18. They want to identify this mutation that's on the X chromosome. Right. Okay. Now, back then, we don't have the kind of high throughput DNA technology like 23andMe, where I can like mail a sample and then
so crazy.
And then like just get back all my DNA results. Right. Back then, this is in the 90s, so this is before Human Genome Project.
Right.
Like this is when in the heyday of that. So we've got these like really, really old technologies. One of them is called BAC cloning, bacterial artificial chromosomes. What you do is you take a segment of whatever part of DNA that you're trying to sequence, you stick that into the bacterial chromosome and then have the bacterial replicate it. And then, you can then start testing each little part. There's also called shotgun sequencing, where you break up the DNA into different parts. You sequence each part, and then you play a jigsaw puzzle where you're trying to see how they overlap. And it's like an intensive process. Requires like 2 or 3 years from like 2 or 3 researchers. Nowadays, it's like a day, $100 and a laptop, right? Like, but back then, this is like several PhDs.
Right.
It's just like figuring out what are these 500 kilobase stretch of DNA doing. And so if you go to photo 19, we've got a photo from their paper. On the top there, that's the 500 kilobase stretch of DNA, okay, from the X chromosome.
Yep.
And they figured out that they've got 20 candidate genes that they need to sequence. So one by one, they're sequencing the 20 candidate genes. And it was their 20th gene.
Of course it is.
Of course it is. They could have given up, but they didn't. They're like, let's just do all 20. Finally, on their 20th gene, they found the mutation that was responsible for this Scurfy mouse strain.
Yes.
Okay, it was a 2-base insertion, and if there's an insertion of 2 bases, remember, it's 3 base pairs to a single amino acid.
Yes.
Right. So if I put in 2, not only have I made a mutation right there, I've also done what's called a frame shift.
Yeah.
Right.
Yep.
Like if you're reading music, like I've just made each measure, like now half of this guy is in the other measure. And so my reading for all of the rest is going to be completely nuts.
Offset. Yeah.
It's not a point mutation.
Right.
And so this 2-base insertion created like a made the gene effectively shorter because later on there was part of that frame shift was like, oh, this is a stop codon, which means I'm going to stop making my protein. Well, the protein is supposed to be so long, so like now it's just completely wrecked, right? It's completely wrecked, and so they named this gene the fork head box protein P3 or FOXP3. Okay, and it turned out that it was a transcription factor, and they published this in Nature Genetics 2001. That was their first big paper with the two of them together. Again, this is Brunkow and Ramsdell.
Yes.
They also published another one in Nature Genetics where they linked this mutation, the Scurfy mice mutation, to diabetes and other autoimmune disorders, specifically IPEX, which is immune dysregulation, polyendocrinopathy, enteropathy, and it's also X-linked. Which is crazy, right? Because like on the mouse chromosome, it was also on the X chromosome, and in humans, it's also on the X. This sort of also shows why it's why we do animal studies in the first place.
Right.
You know, we'd like to think that we're very different, but like, you know, at a real molecular biology level, we're very similar, especially for old systems like the immune system.
That makes sense.
Right? The immune system is a very old system for mammals. So a lot of the same mechanisms are happening across the mammalian species.
Yes.
So the final paper that they published was also in Nature Genetics. I guess the editor there really liked them or something. But
No
all in
reviewer
2001. All in 2001. 3 papers in Nature Genetics. And this was the nail in the coffin. What they did was crossbreeding of Scurfy mice with people that had the wild type, and they could show a recovery of function. And so it was really like it's like definitely that's the location that's causing all of this.
Yes.
And so now we've got a mechanism in the DNA that is perhaps where we've got a location in our DNA where perhaps this regulation is happening.
Yes.
Okay.
Yes.
And this is when Sakaguchi gets back.
Yes.
So in 2003, he publishes this work, which basically combines those two papers. He combines the Sakaguchi paper from earlier, which showed that these T cell regulators were functioning in this way to suppress this autoimmune response.
Yes.
And then it's combining this FOXP3 gene thing that's happened in 2001 to show that actually if you add the FOXP3 gene to normal T cells, they'll turn into regulatory T cells. It's like a retraining.
That's,
you know, it's like you send them back to the training academy
to relearn what it may have been missed.
Yeah, so you can insert this FOXP3 genome, and it'll actually like turn them into regulatory T cells.
That's very okay.
Okay, so it's so cool, like the mechanistic way in which we can manipulate these things to really be convinced that this is how it works.
Yes.
You know?
Yes. No, 100%.
It's really cool. So he published this in 2003 in Science. I think that's 24. You can see.
Yes.
And this is sort of the last of the papers that we're going to review. Okay. And so 2003, and then now it's been what, about 20 years, 20 plus years, and they get the Nobel Prize for it,
which is so.
But Sakaguchi's been at it since the 1980s. Dude.
Right.
He's been at it like for a while.
Right.
That's crazy. Yeah. And so that's effectively what this Nobel Prize is about. It's completely characterizing this military police kind of narrative, or the police of the police.
Yes.
And in photo 25, I have like they always make a cartoon of like what the Nobel Prize is about. So here you've got like you've got the Treg,
the regulatory T cell.
The regulatory T cell, which is like the spaceship, I guess.
Yes.
And it's got the receptor, the T cell receptor that's trying to identify if there's been an incorrect immune response.
Yes.
And then the alien or whatever is inside.
Yes.
That has a FOXP3 cap on. You know, so that's the DNA.
Yeah.
That is I don't know how they come up with this.
That's clever. But that's actually having walked through, because I saw this image at the beginning, it meant nothing.
Yeah.
And now having walked through, I actually can point to each of the things. And
so
identify
each of those,
what's going on and why it matters.
Yeah. So the Treg outside, that's Sakaguchi, and then the FOXP3 gene that is responsible for that is Brunkow and Ramsdell. You know? And so congrats to the 3 laureates.
Yes, yes.
You know, they did amazing work, and now in current frontiers, there's a lot of stuff going on. So
I was actually going to ask, like, so now knowing that we have that understanding, because this was like rather than like the implementation of a tool necessarily like CRISPR, this was more of an understanding of a functional processes in a way that unlocks a whole variety of implications.
Yes, yeah. Now we can start getting into how do we understand when Tregs themselves don't do their job, right? Because that's when we get these autoimmune disorders like IPEX and diabetes.
And the reason it's among other things, the reason it's so important is because it's so directly related to this autoimmune disorder problem.
Exactly. Yeah, this is one of the ways in which an autoimmune disorder manifests,
which is very pernicious.
Yes, exactly,
problem.
And it's something that we need to solve. And at the end of the day, it is a Nobel Prize for medicine, right? And so now there's been so much research about it, dude. Like now there's subsets of Tregs. There's something called the natural Tregs, and then there's induced Tregs that are induced in peripheral tissues after antigen exposure. Okay. So there's like normal T cells that like get induced to become Tregs because of their environment. They decide to go back to their own, because the gene is already in there, right? In their DNA, it's just been repressed.
Yes,
they can take out that repressor, and then you can also induce them in vivo and use them in cell therapy. You know, it's right now we've got work in 2025. I was just reading some of the work that's come out now.
Yes,
there needs to be people have found that in inflamed tissues where there is an immune response, you'll find that the Tregs actually need stronger FOXP3 expression. So they need more expression of that magic gene in order to actually remain stable. They've identified repressors, transcriptional repressors, which are literally like little proteins that come in and sit on your DNA and prevent that stretch of DNA from getting transcribed, right?
Yep.
And so they've discovered repressors that can inhibit FOXP3,
from
right? And then if you block the repressor, then this thing is no longer inhibited, and you can actually get regulation.
Yes.
So that's one way that you can actually do therapy.
Yes.
You know, they've discovered enhancers, which go in and actually do the volume control on that FOXP3 gene that we talked about on another episode.
Yes.
There's like 200 plus clinical trials that are happening right now
related to
Treg.
Related to Treg as a general.
A general so you know, it's a ubiquitous now like method by which the people are trying to tune the immune system and then fight disease in this way. And it wouldn't be possible without like fundamentally understanding all of these mechanisms down to the molecular level of like how this stuff works.
Yes.
You know?
Yes. 100%.
Yeah.
This is a the immune, I mean, obviously, you know, the immune system and all of the medicine in particular, I think it's been post-COVID, there's been a very interesting discussion about how the body works that is not necessarily informed from first principles.
Yeah.
Right? Because there's a lot of these fundamental processes that just are what they are.
Yeah.
And there's a benefit to having an understanding of what those are.
Yeah. I mean, I love thinking about the body as just a bunch of Lego blocks. Right? That are just trying to find each other. And like all of the complicated decision making is happening at that Lego block level, right? At the molecular level. Right. It's kind of insane. Dude, it's such a complicated set of things.
Right.
Where like tiny things go wrong and it just totally wrecks you. Right?
Yeah.
And now we're getting to understanding things at such a level when those tiny things go wrong, we can go in and fix it.
And even just one very, very tiny, small aspect of this combinatorial process that has so many implications. Yeah. So it seems like this has solved sort of this 100-year mystery of like how the immune system restrains itself.
Yeah, exactly. Yeah. Pretty cool.
This is very cool. This is day one, so this is the medicine.
Yep.
I said in our predictions Princeton was going to get on the board.
Yeah, I didn't know it was going to be the first day.
We're on the board.
Yeah, I mean, at least my predictions were wrong, but we got on the board.
We got on the board.
Yeah.
We'll take that. Directionally accurate, but tactically wrong.
Yeah.
So tomorrow we're going to be doing physics. Same time.
Yep.
Same place. Congratulations to the winners. And we're going to, again, make sure we put all these out across all the socials, etc. So be sure to tune in, but we will be back tomorrow for day 2 of Nobel Prize week. My name is Lester Nare, as always, joined by my co-host and our resident PhD, Krishna Choudhary. This is From First Principles. We'll see you guys tomorrow.